Okay, guys, this is the first of what will probably be three video lectures for Chapter 16 on community structure. So, a community is a group of species that inhabit the same area or a given area that interact with each other either directly or indirectly. So, you can think of a community as all the species living in a given area at the same time, and it deals mostly with how they do actually interact with each other, directly or indirectly. So the question then is, what types of interactions occur among the members, the species that live in a community? And what are the characteristics or attributes of a community that do not exist at lower levels of organization, such as the population level or the species level? All right, so the concept of a biological or ecological community is a spatial concept, okay? The species that live in a particular place with a defined boundary. The term can be used more narrowly also, such as the plant community specifically, or maybe the bird or small mammal community specifically. All right, so species in the community will interact in quite a variety of different ways. And these different ways include, first of all, as we've already seen a little bit, competition, okay? Certainly predation, and something we haven't really talked about a whole lot is mutualism, all right? And we'll see more about that a little bit later on. So, what then are the species attributes of a community? Well, we have species number and their relative abundance, and those are important attributes. We have the physical structure of the community. You can think of this as sort of the way the community appears physically, and this is more often than not defined by the major plant growth forms, so the trees, the understory, that sort of thing. And then also, in terms of another attribute of the community, we have the interactions among the species, okay? All right, so let's talk about species composition first. Species composition defines the biological structure of a community. So we can ask, how is the relative abundance of species in a community measured? Well, there are a variety of different ways of measuring the abundance of species, the relative abundance. We can even look at what's called evenness that we'll see a little bit later on. But let's talk about relative abundance first. So the number of individuals of each species in a community can be counted directly, or in cases where we can't count them directly, we can estimate their abundance. We saw that with the marker capture procedure earlier on in the semester. But there you're just counting or estimating absolute numbers of individuals, and in many cases that's not actually the most informative way to get a handle on diversity. A more meaningful measure is called relative abundance, all right? So I'm gonna write that down. Relative abundance. This is simply the proportion of each species relative to the total number of individuals of all the species found in the community. So relative abundance then is P sub I equals N sub I divided by N. So P sub I is simply the proportion of individuals of species I, just a given species. N sub I is the number of individuals of that species, and then capital N is the total number of individuals of all species in the community. All right, so we can look at this in terms of a real-life example, species composition of two forest communities in West Virginia. So here, as you see in this table, and I apologize, there's a lot of data on this table and it's kind of small. Species are listed by rank abundance from most to least. Okay, and you can see that right here. So here's the rank abundance. So we have yellow poplar being listed first because it has the largest number of individuals, or that species has the largest population, 76. Next most abundant is white oak at 36 individuals, then black oak at 17, and so on down the line. Okay. In the next column, in this stand 1, the first stand, we see that relative abundance is listed. And so we have 29.7% of the individuals in this population consisting of yellow poplar, or this community rather, consisting of yellow poplar. How do we arrive at that percentage? you take 76, which is the number of yellow poplar, and divide it by 256, which is the total number of individuals, and you get 29.7%. All right, then you see we have 14.1% white oak, and the numbers get small really quick and go on down from there. Now look in stand 2. So this is a different stand in the same area. We have number of individuals here of yellow poplars 122 out of 274. So that makes up a total relative abundance of 44.5%, quite a bit different than what we saw in stand 1. But then notice there's no white oak, there's no black oak in this second stand. And everything else, with the exception of sassafras, is present at really low relative abundance. Sassafras is 39%, everything else is very, very low compared to that. So you can see just by looking at this table that the structure of these two forest stands in West Virginia are quite a bit different. Okay, so stand 1, again, 256 individuals from 24 different species. Okay, and then stand 2, we have 274 individuals, but distributed only amongst 10 different species. All right, so and also in stand 2, we have 2 species that by themselves comprise 84% of the total individuals in the community. That's going to be the yellow poplar and the sassafras. Okay, so this second stand is dominated by those two. In the first stand, or stand 1, 2 species comprise only about 44% of the total. That's quite a bit different there. And you can see that's going to be the yellow poplar and the white oak. All right, so quite a bit different community structure in these 2 different stands. Okay, so we can look at this graphically through what's called a rank abundance diagram. This is a diagram or a plot that's going to plot the rank abundance on the x-axis against the corresponding relative abundance on the y-axis. All right, we'll see this here in just a moment. But basically, this diagram is going to provide useful information on 2 features of community structure and the total diversity within the community. So it's going to show us something about what we call species richness, or S. So let me write that down real quick. So it's going to show us information about species richness, okay, S, and also what we call species evenness. All right, so take a look at our rank abundance diagram. So again, what we have is our rank on the x-axis. So the lowest number here would be the most abundant species. So for example, we have white oak here in stand 1. And then as you go to the right, their abundance goes down. Okay, so you had species number 24, which I think was represented by 1 individual. Okay, relative abundance, you can see here on the y-axis. All right, so how do we interpret these rank abundance curves? Well, first of all, the longer the curve, and you see we have a longer curve here for stand 1. By the way, the brown here is stand 1, the orange is stand 2. We have a longer curve for stand 1, and the longer the curve, the greater the species richness in the community. Remember, we had 24 species in stand 1. Now, and only 10 in stand 2. Also, the more gradual the slope, the greater the species evenness in the community. In other words, the more evenly distributed amongst the species in the community the individuals of that community are. So here you can see we have in stand 2, 2 species that make up the vast majority of individuals, or most individuals are found in those 2 species, I should say, and then it drops off very quickly. That's not a very even stand. It's dominated by 2 species. Here in stand 1, yeah, we have species 1 and 2 that are fairly abundant, but they don't overpower everything else really, and you see we have quite a few of these species here, for example. So it has a lower slope, or more gradual slope. That indicates higher species evenness. So we can ask ourselves then, what would the curve for a community look like if each species was equally abundant? Well, in that case, the curve would be a straight line parallel to the x-axis. Okay, so it would run parallel to the x-axis like so, be a straight line. And it would have its y-axis intercept would be at 1 divided by S. So again, S is our species richness, how many species we have. And so 1 divided by s would tell you where on the y-axis that line would intersect. So for example, if our community only had one species, then 1 divided by 1 would equal 1, and your y-intercept would be right there. You'd have a line that would run right across the top of the graph like that. If our community had 10 species, for example, the y-intercept would then equal 0.1 or 0.1, right? So 1 divided by 10 in that case, 0.1. All right, so species richness and species evenness are both components of what we call species diversity. So important question to ask is, why is species richness alone not an adequate description of species diversity? Well, as we've already sort of seen, we need to take into account evenness, because you could have a large number of species, for example, in a community, but it's dominated by 1 or 2. And if you're just looking at species richness, the number of species, that might give you sort of a skewed or inaccurate view, I guess you could say, of what the overall diversity in that community is. You need to take into account, how are the species distributed, or how are the individuals in the community distributed amongst the species that are found there. And that's why evenness is an important consideration as well. All right, so how do we do that? Well, we have a number of different ways of sort of defining or, I guess, quantifying species richness and also evenness. And one of the most common ways of doing this, and one of the ways it's been used for quite a long time, is what's called Simpson's Diversity Index. All right, Simpson's Diversity Index. So this actually refers to various closely related indices that consider both species number and relative abundance. So the first related index here under this heading of Simpson's Diversity Index is simply Simpson's Index. All right, we call that D, Simpson's Index D. This is defined as the probability that 2 randomly selected individuals from the community will belong to the same species. So you go out into a community, let's say you're looking at the trees, you randomly select 2 trees. What is the probability that they belong to the same species? That's what Simpson's Index tells you, all right? And so we define that as D equals the sum of P sub I squared. So again, P sub I is the proportion of total individuals in a community represented by the species. It's a relative abundance. And we simply square those and add them all up. All right. And so this value then is going to range between 0 and 1. So Simpson's Index ranges between 0 and 1. All right. Now, the thing about this is that it's counterintuitive, okay? And because this range of values seems to be counterintuitive, where D equaling 1 would mean no diversity, and as D approaches 0, your diversity goes up, it can be a little bit confusing to think about, or to describe or talk about, okay? So that means we have another way of looking at this, which, again, very similar. It's called Simpson's Index of Diversity. This is simply 1 minus D, okay, Simpson's Index of Diversity. And this is a way to get around that sort of counterintuitive issue where a Simpson's Index of 1 means no diversity, and a Simpson's Index of 0.1, let's say, would mean a lot more diversity. Again, that's sort of counterintuitive, so the Simpson's Index of Diversity, 1 minus D, helps us to get around that. In this case, the value increases with species diversity. This index represents the probability that 2 individuals randomly drawn from a community will belong to different species. Okay, that's what it tells you. The probability that 2 randomly selected individuals do actually belong to 2 different species. All right. Next, we can also calculate evenness. All right, so we've talked about species diversity with Simpson's Index, for example. Now we can look at what's called evenness. And so evenness can be calculated as E sub D equals 1 over D divided by S. All right, so the value of evenness ranges from 0 to 1, and at 1, all species are equally abundant. So we take 1, divide it by our Simpson's index, and divide that by our species richness, S. And that gives us an index or a measure of evenness. And again, at 1, all species are equally abundant. All right, now, in a lot of cases, Simpson's index is not actually used. There are a lot of ecologists, for whatever reason, for various reasons, will actually use something a little bit different, what's called the Shannon index or the Shannon-Weiner index. So I'll just write that over here, for example. All right, so I'll have the Shannon index. This is a commonly used metric as well, sometimes called the Shannon-Weiner index. Either name is fine. This also considers species richness and evenness, okay? And so we symbolize it as H. All right, so what is our formula here then for this? Well, our Shannon index equals the negative sum of P sub I times, screen's touchy, times the natural log, that's ln, natural log, P sub I. Okay, so again, P sub I is the proportion of the total individuals in the community that's represented by the species by species I, okay? ln is the natural logarithm. And so when we do this calculation, we get a minimum value of 0. That would mean you have one species present. So if you had one species only in your community and you did this calculation, you would get a Shannon index value of 0. The maximum value is the natural log of the species richness. It's the natural log of S. That would be all species being present in equal numbers in that case. Okay, so you see how this then gives us Shannon index that includes both species richness and evenness. It's viewed as being a little bit more robust than Simpson's index. It provides more information, and that's why a lot of times people use that rather than, or use Shannon index rather than Simpson's index. Okay, so this maximum value, what we call H max, where that equals our species richness, that can be used to calculate an index of species evenness. All right, and so when we do that, we get this index of evenness, E sub H, which equals H over H max. All right, that value ranges again from 0 to 1. 1 would be complete evenness with all species equally abundant. So you can see there are a variety of different ways here of sort of quantifying or getting at species richness and also species evenness. Okay, let's shift gears now and talk about dominance in a community. So when a single or few species only, let's say, predominate in a community, those species are referred to as dominants, okay? They are dominants in terms of their numbers, possibly the effect they have on other species in the community and that sort of thing. So dominance, if you think about it, is the converse of diversity, okay? The less diversity you have, probably the more dominance you have in terms of one species dominating the others and vice versa. So we can ask, do dominant species always have the most biomass, let's say, within the community? Are dominant species always the most important species in terms of the way that the community functions, right? Probably not in a lot of cases, or even in most cases. So the basic Simpson's index that we looked at, D, is often used as a way to measure dominance. So let's say, for example, D equals 1. In that case, we only have one species present. So that one species is what we would call completely dominant, and there's no diversity in that community whatsoever. Dominant species are most often defined separately for different taxonomic or functional groups as well. So, for example, we may have a community they'll have both a dominant tree species and maybe a dominant small mammal species or a dominant bird species or something like that. So we can talk about this in terms of different different groups of species or functional groups or taxonomic categories within the community as well. All right. Now, abundance alone is not, in most cases, a sufficient measure of dominance. So in a forest, for example, there may be more small understory trees, saplings and things like that, and more juvenile trees. But the fewer larger trees may actually have the most biomass. So you can have a lot of really small understory trees, but because they're small, they don't add up to a lot of biomass. You may only have a few really large adult trees, but because they're so massive, most of the biomass actually is tied up in them. And we can actually see this when we look at some data here in a moment from a deciduous forest in Virginia. So go ahead and take a look at that. So we have this table here, 16.2, structure of a deciduous forest stand in central Virginia. And here we can see that the relative abundance, 60% of the trees are red maple and dogwood. Okay, red maple and dogwood, or not quite. Yeah, it is actually. Okay, so we have here relative abundance. You add them up, you get about 60%. Relative biomass, however, 60% of that is tied up in white oak. All right, so take a look at the relative biomass. Very little actually in the species that have the highest relative abundance. Most of the biomass, nearly 60%, is tied up in a species that only has the third highest relative abundance. This tells us that these white oak trees are probably older individuals that are very large, and those dogwoods and red maples probably make up much of the understory of saplings and trees that are relatively young, and that's why the relative biomass is so little. So that shows you that it's important to look at a number of different sort of metrics in order to try and get a handle on the structure of a community, in this case, a forest, right? Different species in the community may have very different relative abundance compared to relative biomass, and so it pays to look at it in different ways. All right. Next thing let's talk about in this first part of the video lecture is keystone species. So a keystone species is just a species, or in some cases more than one species, that has or have a disproportionate impact on the community relative to its abundance. So we're talking about a species here that's not very abundant maybe, but because of what it does or a role that it plays in the community, it can have a disproportionately large impact on a wide number of other species, or a large number of other species in that community. So how can a species then exert strong influence on its community when it's less abundant? This is a key point to understand. We'll see some examples of this in a moment. And then also we can ask, what effects can the removal of a keystone species have on a community? And that second question there is of central importance in conservation biology and conservation of endangered species. Removing a keystone species can have really wide-ranging and rippling effects throughout the overall community, which, depending on the species we're talking about, might cause that community to collapse. So keystone species function in a unique and usually significant way within a community. Their effect is much greater and relatively disproportionate to how abundant they are numerically. All right, so the role of a keystone species may be to create or modify habitats, for example. It could influence interactions among other species. The removal of the keystone species as a result of that could lead to changes in the structure of the community, steep declines in biodiversity because that keystone species was so integral to creating maybe habitats, let's say, for other species. Great example of that is a species of coral, Oculina arbuscula, which lives off the east coast of the US and Canada. This is a keystone species. It's the only coral in the region with a structurally complex branching shape. So think of corals that kind of look like deer antlers in terms of their overall structure. So because of its shape, it creates habitat for upwards of 300 different species of invertebrates that actually live among those branches of that coral. And many of those species actually complete much of their life cycle within the coral. So they're absolutely dependent on the existence of that coral for their own habitat. All right. Also, another example of this, keystone herbivores can actually modify the community through their feeding activities. African elephants are a great example of this in the savannas of southern Africa. These guys feed mainly on woody plants, so the browsers, as you can see here, this elephant is doing. And they're very destructive feeders. They often topple trees, uproot them, breaking and destroying the shrubs they eat. They're obviously very powerful animals. And this reduction then of tree and shrub density due to the elephant behavior actually favors growth and reproduction of grasses. So this community change then brought on by the elephants and their behaviors benefits grazing herbivores, but it actually doesn't benefit the elephants. Okay, so this is something that has this behavior, I guess I should say, of the elephants has ripple effects throughout the other herbivore assemblages within that community. Some field studies in Kenya found that grass productivity and large herbivore density increase with decreasing tree cover. So it could be that there's a link here between the elephant's behavior and how well or how productive grasses are, which certainly then would impact the herbivore density that feeds on those grasses. And we can see that, some of that data here in this figure. So what we see on the x-axis here is percent tree canopy cover, so from low to high. So we have less dense canopy, higher density canopies here. We have grass biomass, so mean number of pin hits. That's just a way of measuring grass blade density. Don't worry about the method there, not important. And we can see here we have two measures of this below tree canopies and between tree canopies. Now, the author of your book is suggesting that the slope of this line is significantly different from zero. Based on the look of that, I would have to argue that that's probably not the case. I can't imagine that slope being anything other than zero, but I don't have the data to look at, so I don't know. But let's look at the orange line, which is below tree canopy grass density. We can see that you have the highest grass density at low tree canopy density, and as density goes up, or canopy density goes up, you can see that grass biomass goes down. Well, it makes sense then if you think about the elephants, feeding, browsing in the very destructive way that they do, thinning out the tree canopy or maintaining it at very low densities, for example. If they're keeping tree canopy down here, low densities, then the grass biomass might be higher as a result, and then that could certainly have a positive impact on large herbivore abundance. And that's what this figure shows. Here we have a number of trees, so another way of measuring tree density, basically tree canopy density, from low to high. And you see at the lowest tree densities, we have the highest herbivore abundance. And presumably that's because the grass biomass is so much higher at low tree canopy density due to elephant behavior. So elephants, as a result of this and other kinds of interactions, are viewed as a keystone species. Elephants were to go to extinct, for example, which they're certainly in danger of doing because of poaching and whatnot. You can imagine that the tree densities and canopy densities might go up, reducing, therefore, the grass biomass, which could then have a negative impact on herbivore abundance. All right, so destruction of trees by elephants creates habitats also for smaller vertebrates. So this is something we haven't talked about yet. We've been talking about large vertebrate herbivore density as a result of a trickle-down effect of feeding behavior of elephants. But what about other small vertebrates, for example? Well, there was a study in central Kenya that found that lizard density actually increased with the density of trees damaged by elephants. So the more tree damage the elephants were doing, the better that seemed to be for lizard populations. Another study in the Eastern Cape region of South Africa found a similar effect of elephant feeding on community diversity. This one looked at sort of a grass and bird species diversity. They found that sites inhabited by elephants had an increase in both grass diversity and species diversity, bird species diversity rather, compared to habitats that did not have elephants. So the effects of elephants as a keystone species can be fairly wide-ranging and can be quite unpredictable and surprising in a lot of cases. And that's the real danger of having a keystone species being removed from a community through extinction or whatnot. You just don't know what the ripple effects might be. All right. Predators can also be keystone species in communities. Great example here is sea otters, killer whales, sea urchins, and the kelp beds of the Pacific Northwest. In the 1990s, and I'm not sure what the reasons were for this, but in the 1990s, there was a documented decline in sea otter abundance due to increased predation by orcas or killer whales. And that had ripple effects on the overall kelp community, which we're going to describe here in a moment. So kelp beds are fairly diverse communities, marine communities. They're a habitat for a wide variety of different species, and sea urchins are one of those. Sea urchins feed directly on the kelp, okay? Sea otters feed on sea urchins, and then in turn, the sea otters are fed upon by killer whales. So in the 1990s, when sea otter populations declined as a result of killer whale predation, the sea otters, which are viewed as a keystone species, were effectively being removed from these kelp bed communities. That resulted in an explosion in the sea urchin population, and the sea urchins then subsequently overgrazed the kelp, reducing habitat for other species. So as a result of this sort of chain reaction of events, overall species diversity in the community, the kelp bed community, declined because the sea urchins overgrazed the kelp. And the kelp is essentially like the tree community of a forest, for example, habitat for a wide variety of different species. And this is what you see here in this figure. So I'm going to kind of zoom in on this because it's kind of small. There's a lot going on here. So you see on the left-hand side over here, we have sea otters feeding on sea urchins. Sea urchins are shown here in purple. And so they're keeping the sea urchin populations fairly low. And that means that because sea urchins are fairly low in density, the kelp is not being overgrazed, so you have a healthy kelp forest shown here. All right. Now, when the killer whales began increasing, when we started to get increasing predation pressure by killer whales in the 1990s on sea otters, sea otter populations were reduced. You see that the sea urchin population as a result sort of exploded, and as a result, they overgrazed the kelp. And you can see all of this here in these figures. Here you can see the sea otter abundance declining, especially over the 1990s, that decade there. And again, I'm not sure what the reason for that was. I'm sure studies have shown why this happened, but I'm not familiar with it. And then what happened with sea urchin biomass as a result, you can see in 1997, it peaked. And here we're talking grams per quarter meter squared. So we're talking a lot of sea urchin biomass here compared to what might have been usually the case. And then you see that resulted in a definite increase in grazing intensity on the kelp and resulted in a drastic decline in overall kelp density. So this is a classic example of how removal of a keystone species, in this case a sea otter, can have lasting ripple effects that spread out through the community and overall affect other species of vertebrates and invertebrates as well. This kind of kelp habitat that's been overgrazed is not going to support fish. It's not going to be a good nursery, which kelp habitats very often are for other species. And so you can imagine how overall species diversity would decline. Okay, so I think I'm going to cut off here for this first video lecture. We'll start up next with the second part of this for the next video lecture.